It could go in many different directions. So I have done my best to kind of break this down into an understandable chunks. And I've also done my best to keep this very lay audience appropriate. So please don't hesitate to ask questions or ask me to clarify anything. I think we'll have plenty of time at the end. So fibrous dysplasia, McCune-Albright syndrome is a very complex mosaic disorder due to activation of a protein called G-alpha-S. And probably most folks on the call are familiar with the fact that this is a disease that can involve many different systems, but in particular, the bone, endocrine, and skin systems. So the essential problem in fdmas is this G-alpha-S receptor. This is a hormone receptor, and here you can see its overall structure. This is the membrane of the cell, and the receptor has parts that span all the way from the outside of the cell to the inside of the cell. And the way it works is a hormone will bind to the receptor, and then the receptor transmits the signal through the membrane, and then it comes out the other end, and then this leads to increased production of different types of hormones. So in the mutant state, this G-alpha-S normally has the job of turning the receptor off and on, and instead it's stuck in the on position. So essentially we have too much production of the different hormones that signal through the receptor. And which hormones are produced depends on which organ system the receptor is located in. So if the receptor is in the cell, in the skin, we get too much melanin production, and that leads to skin pigmentation. Sex steroids signal through this receptor, so we can get precocious puberty if the receptor is in the sex organs. We can have too much growth hormone, too much thyroid hormone, etc. And then just briefly as a little bit more background, fdmas is mosaic, so it only occurs in some parts of the body. It never occurs in all of the parts of the body. And that's because the mutation that causes this disease happens during embryogenesis while a baby is still forming. Very early in embryogenesis, the mutation happens by chance. And then everywhere that cell spreads throughout the developing embryo, it creates tissues that are affected by disease. So because we have disease in multiple different tissue organs, we know that the mutation had to take place very early before these different tissues differentiate. In bone, G-alpha-S does something a little different. It impairs the development of bone cells. So the cell type we're most interested in is called a skeletal stem cell, and it can turn into a bunch of different types of cells, including an osteoblast or a bone building cell, a cartilage cell, a fat cell, or a marrow cell. And all these different cell types are important for the bones. When a G-alpha-S mutation is present, all these different pathways get blocked. And instead of turning into all these different cell types, the skeletal stem cell can only form an abnormal osteoblast or an abnormal bone building cell. And it's the messed up activity of those abnormal osteoblasts that leads to all the problems in FD. So here you can see a normal bone, a picture of a normal bone under a microscope, and compare it to FD tissue. And in normal bone, you can see all those different cell types. We have nice marrow cells, nice fat cells, and we have this very nice, beautiful, organized bone that's formed. In FD, instead, we have this big mass of fibrous tissue. And what this fibrous tissue represents is a bunch of those abnormal osteoblasts that are trying to turn into different cell types, but they can't. They're basically stuck in this stage of development. So they divide and divide and divide, and they fill up the marrow cavity. They can create some bone as well, but the bone they make is very disorganized, and it's not nearly as strong as normal organized bone. And then, again, I won't spend too much time on this, but it's the formation of that disorganized bone and that extra fibrous tissue that leads to all the problems we see in FD. When FD involves the skull, the growth and the expansion of the fibrous tissue can lead to facial asymmetry, vision and hearing problems. When it affects the spine or the ribs, the bone is weaker, it can bend, leading to scoliosis and fractures. And the same thing can happen in the limbs. We can have fractures, bowing, difficulty walking, and in all of these compartments, fibrous dysplasia can cause pain. So for the purposes of this talk, I'm going to focus on 3 possible strategies that people have tried to to treat FD. So these are the kind of the main obvious mechanisms we could go after. So here's our FD cell. And one way that FD causes problems is these cells can release signals that can alter the behavior of other cells. So one possibility that a lot of people have tried is to try to change these signals that are coming out of the cell and force the FD cell to act more like a normal cell. Another possibility focuses on that receptor that's signaling too much. Is there some way to change the activity of this receptor? Can we turn it off, or can we prevent it from signaling so much? And then the third strategy I'm going to talk about is, can we actually fix the DNA in the cell itself? Can we take that abnormal gene and just turn it back and change it and turn it into a normal gene? And the first strategy I'm going to talk about is changing the signals that are released by the FD cells, because this is where lots of work is going on right now. And this is where I think we have the potential to have therapies that are really going to impact people in the short term. So there's 3 strategies people are using to try to change these signals, and these are the names of the medications that are associated with it. So there's a protein rankle that FD makes. We can target that with a drug called Denosumab. FD makes a protein called IL-6, and there's been attempts to try to treat that with a drug called Denosumab. And finally, FD makes a hormone called FGF23, and then there's strategies to try to treat that with a drug called burosumab. So first, rankle. So rankle is a protein that controls the balance between bone formation and bone resorption. This is a process that is going on all the time in both FD bone and in healthy bone. So osteoclasts, or bone breakdown cells, they chew up the bone, and then the osteoblasts, or the bone forming cells, come in and make new bone and fill in that hole. And this cycle goes on and on and on, and it's how the bone renews itself and stays healthy. One of the problems we have in FD, though, is this whole cycle is just turned up to 11. This process is happening too fast, too often, and that's part of why the bone that FD makes is so disorganized. So Denosumab, or a rankle inhibitor, one of its goals is to decrease the activity of these osteoclasts that are chewing the bone up, hoping that if we do that, we can bring this whole cycle down, slow down the bone turnover, and help the body build better bone. So there's been lots of interest in using Denosumab as a potential treatment for FD, and the data I'm going to show here, it's pretty hot off the presses data from a trial that we did at NIH in adults. So we gave high-dose Denosumab once a month for 6 months, and we saw we were very encouraged to see that it had a massive effect on that bone turnover cycle. So we can see that in the blood because we checked levels of both bone formation and bone resorption. And you can see these levels were very high when patients started Denosumab, and then they came down dramatically over 6 months of treatment. We also did bone scans to look at how active the FD lesions are. And here again, we had a huge impact. So on these scans, this is called a sodium fluoride PET CT. Patients are injected with a little bit of tracer that goes to the bone. And essentially everywhere you see black is fibrous dysplasia. And the blacker it is, the more active or the more turnover is happening in the bone. And we saw that compared to pre-treatment, there are just huge decreases in the amount of tracer that was being taken up, which is really a wonderful sign that the FD lesion activity is going down. We also saw improvements in pain scores, and patients had improved physical function. However, I'll come back to this later in the talk, but this was an open label study. Everybody got treatment, so it's really difficult to know if you're truly having an effect on pain and physical function without a placebo group. So in this study, we also did bone biopsies before and after treatment because we thought it was very important to understand what impact Denosumab is actually having in the tissue itself. And again, this is where we saw really exciting results. So this was only 6 months of treatment, a very short period of time. But what we saw is that compared to the baseline samples, we were seeing a lot of bone forming within these lesions. So here you see this is just a mess of fibrous tissue. There's very little bone tissue. And then after 6 months, all of this kind of pink blobby stuff, this is brand new bone that's being formed inside the FD lesion. We also saw that the FD cells were not proliferating as much. They weren't dividing as fast. We can see that here because we used marker to stain for cell activity. So everywhere you see a little brown spot is a cell that's turning over and dividing and making more cells. And after denosumab treatment, there was much less cell division. And we also saw increased expression of proteins that are involved in mineralizing bones and making bones stronger. So here, everywhere you see these purple dots, that's a mineralization protein. And you can get a sense that there's much more of it being expressed after denosumab. So altogether, these are beneficial changes that will hopefully make the bones stronger and less active. But there are downsides of denosumab. This is a drug that has some significant potential side effects. The biggest one is bone turnover rebound. So I shared that FD bones are very active. They're turning over a lot, making new bone, breaking bone down over and over again. When we give denosumab, all that turnover comes down beautifully, which is great. But you do pay a price for altering bone turnover, because when this drug is stopped, the bone turnover increases, and it doesn't just go back to where it was before treatment, it goes even higher than it was before treatment, before settling down to right about where it was before. So the most obvious impact of that is that you lose all the benefit for the drug when you're not taking it. But what can also happen, particularly if you have very active FD, is that bone turnover can be associated with high blood calcium levels. So all that new bone that's turning over, it's so active that it's actually dumping pieces of the bone, which includes calcium, into the blood. And that is what we saw on our study. There was one patient who had really severe FD. 3 months after she stopped denosumab, she developed really severe high blood calcium and had to be hospitalized. Fortunately, we were able to treat her well with medication and her calcium levels came down. But what this really pointed to in this study is that we have to be very careful about denosumab use, and especially if patients are young and if they have a lot of FD. There are other safety issues to consider as well. While you can have high blood calcium after denosumab, while you're on denosumab, you can have low blood calcium levels. Bringing down all that bone activity can decrease the amount of calcium that's coming out of your bones, and that can be dangerous too. This is something that's generally treatable as long as you take calcium supplements when you take denosumab, and that you're under the care of an endocrinologist that knows how to manage calcium issues. Osteonecrosis of the jaw is a dental complication that can occur with any medications that impair bone turnover. And there's a this seems to be something that is more often associated with denosumab than with other medications. Also, a very important consideration is that denosumab isn't just going to affect FD, it's going to affect normal bone too. This is a mosaic disease, so most patients have a mix of FD and normal bone. And there's no way to target the denosumab just to the FD. So we can see high bone mass developing, and it's possible that if denosumab is continued for years and years, we may end up seeing an increased risk of fracture in normal bone. So these are all things that still need to be worked out. So where are we now with denosumab? I think there's very exciting potential here to improve FD lesion activity, to make bones stronger, and to prevent a lot of the problems we see in FD. However, this severe post-discontinuation calcium issue is a very important potential safety issue. And there's just a lot of questions that we still don't have the answer to. In particular, who are the patients with FD that should be treated with denosumab? Clearly not everybody, but what are the conditions? Which patients will it be the most effective in? How long should we treat patients for? This is really not known, especially because when the drug is stopped, we lose all the benefit of denosumab. And what dose should we use? We used a very high dose in our adult trial, and it's likely that we may have seen less complications if we used a lower dose. So that's something we still need to answer. So for right now, my thinking on this is that denosumab is a reasonable option to consider for patients who have FD and very severe morbidity or a lot of skeletal complications that aren't responding to more conventional treatment. However, we have to be particularly careful if patients are young and if patients have a lot of fibrous dysplasia. And further study is needed, particularly in children. And I just wanted to talk a little bit about denosumab use in children, because this, I think, is a very exciting potential use of denosumab. So we know that FD lesions are usually not obvious when a patient is born. The lesions expand over time. They usually show up in the first few years of life, as you can see here. They grow and expand while kids are growing. And then right around age 15 or so is when you have the full amount of FD you're ever going to have. So adults don't develop new lesions, and adults shouldn't see substantial growth in existing lesions. So with this makes me wonder is, do we have a window of opportunity here during childhood? If we could treat with a medication that will stop FD lesions from forming and expanding, that could potentially have a really profound effect on someone's disease course. So we are currently conducting a clinical trial of denosumab in kids with FD at NIH. This is going to enroll 15 children ages 4 to 14. And the strategy we've taken for this study is to use a very, very low dose of denosumab. Compared to our adult study, this study has 12 times lower of a dose. And we did that because we're expecting that we're likely going to need to continue the denosumab for a fairly long period of time to maintain those benefits. So right now the study is enrolling at NIH. So if anyone has any more interest in it, please feel free to email me. All right, switching gears a bit to another possible.
Sorry, Alison, can I just, and maybe we have a question here, and before we move on from denosumab, about what the age ranges were for the hypercalcemia data.
Yeah, so hypercalcemia, in the study that we conducted here, this patient who developed hypercalcemia was in her early to mid 20s. And there's been 2 other cases of hypercalcemia reported in children. There was a larger retrospective review of denosumab from the Dutch group. So they treated adults clinically, and they did not see hypercalcemia. So I think the difference in the patients that we're treating and the patients that they're treating is that we tend to have more severely affected patients, and that our patients are younger. I don't have a great answer. I can't tell you if there's like one age at which you don't have to worry about hypercalcemia anymore. But I do think particularly patients in their in childhood, in their 20s, and maybe even in their early 30s are some of the ones I'm a little more concerned about.
Thank you.
And that probably reflects the fact that bones are just more active in younger people compared to older people.
Thanks.
Any other questions about denosumab before we move on? And we can always come back to this at the end too, if people have questions. All right, so I wanted to talk about interleukin-6. This is another protein that FD cells have been shown to release. So IL-6 does a lot of different things in the body. It's involved in metabolism, your immune system, muscle function, brain function. But the reason people are interested in it in FD is that it's also involved in bone, and particularly in osteoclasts, or those bone resorbing cells that we've been talking about. So tocilizumab is a drug that binds IL-6 and decreases its activity in all of these areas. So really one of the bigger papers that's come out in FD over the past year is the results of the TOCIDES trial. So this was conducted by a French group, and it was a randomized placebo-controlled trial of tocilizumab in patients with FD. They had a really nice design to the trial. So for the first half of the trial, they took 8 patients and treated them with tocilizumab, and they took 8 other patients and treated them with placebo, or a fake drug. And neither the patients nor the investigators knew who was getting the drug and who was getting the placebo. And then after, I believe, 6 months, they crossed over. So all the tocilizumab patients went on placebo, and all the placebo patients went on tocilizumab. I'll just plug here that placebo-controlled studies that compare patients getting a drug to patients getting a fake drug are the gold standard way of studying medications in medicine. It's a very time-consuming, expensive, complicated way of doing a study, but it's really the best possible way you can do research. So I admire the investigators a lot for doing this. In particular, placebo-controlled studies are the only way we can really determine if drugs have an effect on bone pain. So these were their results, which were very nice, but also very disappointing. So they didn't see any change in bone turnover markers between the tocilizumab and placebo groups. They saw no clear change in pain. Now, I'll say, actually, the patients reported a huge change in pain. Both the tocilizumab group had a 40% decrease in their pain scores. However, what makes this disappointing is that the placebo group also had a 40% decrease in their pain scores. And that is a well-known complication of medicine. The placebo effect can be very profound. And that's one reason why, in particular, in our Denosumab trial, I take the pain scores with a real grain of salt. I think there's no way to know if those are truly related to Denosumab without a placebo group. But taking this data together, unfortunately, there's just no evidence that tocilizumab is effective in FD. So disappointing, but it's also wonderful for our community to know that this is not effective and that we can stop treating patients with this. So the third and final kind of cell signaling approach I want to talk about relates to a hormone FGF23, or fibroblast growth factor 23. So this is a hormone that's made by FD bone. Just like endocrine glands make too many hormones, FD bone makes too many hormones. So FGF23 has kind of two main actions. It acts on the kidney and causes it to leak phosphorus into the urine, and it acts on the gut to prevent it from absorbing as much phosphorus from food. So the net effect of these two things together is it decreases the level of phosphorus in your blood. And this is very important in fibrous dysplasia in particular, because we have lots of evidence that low blood phosphorus levels make FD bone weaker. So we see that compared to patients with normal phosphorus levels, shown here in yellow, patients with low phosphorus, shown here in black, have more fractures, and they start having their fractures younger. They have more complications with skull FD, in particular, complications that can happen at the very base of your skull where it joins to the spine. And we see higher risk of scoliosis. And this is all research that has been done on our natural history study, looking at hundreds of patients over the years. So treating FGF23 excess and low blood phosphorus levels is really important in patients with fibrous dysplasia. The problem is this has been one of the most difficult endocrine features to treat. And that's because for years, we just haven't had a good way to affect FGF23 levels. So basically what we have is a leaky bucket, body is spilling phosphorus out into the urine, and our approach has been to just try to fill this leaky bucket. We'll try to dump in phosphorus faster than the body can leak it out. And we do that by giving oral phosphate 3 to 5 times per day, and also by giving a special form of calcitriol that helps you absorb even more phosphorus from your gut. The problem with these medications is they're really inconvenient. 5 times a day medication is just really difficult to be compliant with. Also, they have GI and kidney side effects. So in general, you can't fully correct the blood phosphorus level. The most you can hope for is to maybe squeak it into the very bottom of the normal range. But over the past few years, there's been a new type of treatment for FGF23 excess, and this is with a drug called burosumab. And I apologize, this is kind of a dumb picture, but it was the most simple picture of an antibody I could find. So they're stuffed animals called giant microbes. But this little Y-shaped guy is burosumab, and this little rectangle is FGF23. What burosumab does is it goes in your body and it binds that FGF23 so that it can't act. So instead of filling a leaky bucket, we're trying to stop the bucket from leaking completely. And burosumab has been studied in 2 other diseases where we see high FGF23. One is called X-linked hypophosphatemia. One is called tumor-induced osteomalacia. They're both very different than FD. But one thing they have in common is high FGF23 levels and low blood phosphorus levels. And what's exciting is in these studies, burosumab has been able to fully collect phosphorus. It can get it right up into the middle or even the upper part of the normal range, which is a huge advantage over conventional therapy. And it's also been shown to help fracture healing in those disorders. So burosumab is safe. Unfortunately, it's extremely expensive. It's about $200,000 per year compared to phosphate and calcitriol, which is really cheap. And probably the biggest barrier to its use in FD, it hasn't been studied in FD patients, and the FDA has not approved it for use in FD. However, we're also performing a clinical trial of burosumab right now at NIH. My hope is if we can show it's safe and effective in this study, then this will make it more widely available to the patient community. So this will be 15 patients, and it's a mix of kids and adults, and it's also enrolling now. So if anyone has questions, please feel free to email me. All right, so we talked about how to change signals that are released by FD cells. And now I'd like to switch gears and talk about this mutated receptor. Is there any way to change the receptor, the activity of this receptor that is turned on and signaling too much? There's been a lot of interest in using this as a potential treatment strategy, and we have some good reasons to think it will be an effective one. First off, the mutations that happen in G-alpha-S are really consistent. There's really one spot in the genome where they happen, and there's only two possible ways it can be mutated. So because it's so consistent, that makes it a really attractive possibility to try to develop a drug against it. Also, this G-alpha-S protein is actually targeted in nature. So if anyone's heard of the disease cholera, that is an infectious disease where the bacteria makes a toxin that turns on G-alpha-S. And in that disorder, patients get a lot of diarrhea. So we obviously wouldn't want to replicate that, but we've shown that nature has figured out a way to bind the G-alpha-S protein. So that gives us hope that we may be able to develop a drug that will do the same thing. And also there have been other disorders with similar receptor problems that have been successfully targeted with medications. So there's a lot of different ways you can come about developing a drug this way, but the overall approach that most people would use would be to take a lot of different compounds and test them in model systems. So we have cells that have the G-alpha-S mutation. We have animals that have the G-alpha-S mutation. If you take a large number of compounds and screen them across these models and look and see if they're having any impact. Then when we identify compounds that might be effective, we can go and use medicinal chemistry to look at their structure and see, is this a compound that has any prayer of being turned into a drug? You can then take that altered compound and test it further in cells and animal models, and then that can ultimately lead to drug development. So this is an approach people have been working on for a long time. At NIH, we spent 10 years working on this approach. We tested over 300,000 compounds and unfortunately failed to identify any drug candidates. However, I don't think all hope is lost. This technology is improving all the time. I know there's a number of other groups who are working on this approach and that have access to newer technologies. So I do think this is something we may see down the line. However, it's going to be a long time. Developing a new drug is often a decade-long process. So realistically, this isn't going to be anything we see anytime soon, but it could be a fast-moving area kind of in bench science. All right, and then the last strategy I want to talk about is probably the most intuitive one. Patients ask me about this all the time. Can't we just fix the gene mutation in the DNA in FD? And this is a broad area called gene therapy. Again, there's a couple of different ways you can approach it, but this is a pretty standard way of thinking about it. So we have a cell that contains a mutated gene. Can we put in either a correct gene that will replace this mutated gene, and then that will lead the cell to function normally? So gene therapy is something where we have to figure out a way. It's not enough to just make a new gene. We have to figure out how to get it into the nucleus of the cell where the DNA is contained. And that's done by using what's called a vector. And there's a lot of different ways you can do that, but the most common is to use either a virus, a bacteria, or a plasmid, which is kind of something a virus makes, in order to kind of infiltrate the cell. There are a lot of challenges in trying to come up with the right vector. You have to make sure that the genes actually get to the right place. not only in the right cell, but in the right place in the cell, and then in the right place in the DNA. There's a lot of places on that path that can get disrupted. You also have to make sure you're not having an effect on other genes. And then probably the biggest barrier, practical barrier we've run into is the immune response. Your body is designed to try to avoid this process from happening. Viruses and different pathogens have tried to figure out how to get in and mess with our genes for years. So the body is built up with a lot of defenses against this process. And this is something that has unfortunately really affected us in the gene therapy field. So I don't know if anyone here has heard about this kid, Jesse Gelsinger. So he was an 18-year-old man who had a genetic form of liver disease. And in 1999, he participated in a clinical trial at the University of Pennsylvania, where they were trying to fix the genetic mutation in his liver using gene therapy and an adenovirus vector. And unfortunately, 4 days after he got the injection with gene therapy, he had a massive immune response and developed multi-system organ failure, and he died. So there were a lot of investigations into what went wrong here. And they did show that this trial had a number of ethical and regulatory issues with the way it was being conducted. However, due to both that and due to concerns about gene therapy safety, all of the gene trials that were going on at University of Pennsylvania were suspended. The FDA started looking much more carefully at gene therapy. And overall, it was about a 10 to 20 year setback for the gene therapy field. This was really sad and really unfortunate. However, I do not think all hope is lost. There has been a lot more interest, and recently more trials have been conducted, and they have been done better and more safely. And some of these trials have shown real promise in treating a number of different conditions, including severe combined immune deficiency. This is something that used to be known as bubble boy syndrome, where you essentially have no immune system. So it can really be a life-saving treatment strategy. It's been used in leukemia. It's been used in a genetics form of blindness and in hemophilia. So right now, gene therapy is only available in clinical trials, but I do think this is a space to watch, in particular because we have had real improvements in gene editing techniques. CRISPR is something a lot of patients ask me about. That is a really great technology that it can allow you to very precisely cut the DNA and change the genetic makeup of the DNA. So that's a real potential advancement. It doesn't solve the vector problem. I still think we need a lot of work to find the right vector to get the CRISPR system into the cell, but it's really exciting. I think overall, though, for me personally, I would have to see extensive use of gene therapy in life-threatening disorders before I'd be willing to use it for fibrous dysplasia. As disabling as fibrous dysplasia can be, it's very rarely life-threatening. So I think we have to be really convinced that these are safe and effective strategies before we start using them in our patients. And then this is my final slide. I just wanted to very briefly mention the possibility of stem cell therapy, because I get asked this quite a bit. And this is also a potentially intuitive way to treat FD. I showed you on an earlier cell that the G-alpha-S mutations in FD disrupt the activity of skeletal stem cells. So one possibility to treat it could be to just aspirate out normal skeletal stem cells, grow them in a dish, remove the messed up bone, and just inject the normal cells into the bone and then have it heal up. And I think this makes a lot of sense logically, but the actual application of this is really, really difficult. The biggest barrier is there's just no way to make stem cells stay where you want them to go. I know there are a lot of orthopedic surgeons and other practitioners that will advertise using stem cell therapy. They'll inject stem cells into joints and in other places. The problem is the stem cells will get taken up in the blood and they can go all over. There's really no good proof that they stay in the bone. Also, even if they did stay in the bone, the stem cells don't necessarily know how to form a 3D structure. For bone, it's really important that it's formed precisely. We want it to be exactly the right width and exactly the right length. And a lot of these cells don't have the right programming to know how to do that. So I don't think this is currently a feasible therapy for FD. I don't have a ton of hope for this strategy, but I may be wrong. You never know what could happen in the future. So that is it for me. I am happy to take any questions.
Thank you, Alison. Yeah, at this point, if our folks who are on Zoom with us, if they care to join us, I welcome that. And then you can ask your questions more directly. As folks are doing that, though, we did get quite a few questions beforehand. So some of them you've addressed: CRISPR, gene therapy. Those are things that keep coming up. Additionally, we have some questions about mouse modeling. You know, we've developed in the fdmas field some mouse models, and are those still being widely used and developed?
Yeah, it's a great question. The mouse models in FD have been a real success story, I think. There's several different models that have been developed over the past about 10 years, and most of them are in wide use. In fact, I didn't have time to share that in this research update, but they've informed in particular our Denosumab work quite a bit. Every single thing we did in the human study of Denosumab, we also did in mice, and we saw very, very consistent responses. The nice part about a mouse model is you can do much more detailed analysis of the bone tissue, so it's really informative. And I think the presence of the mouse model gives me a lot of hope that as we develop more treatments, we're going to have a great system that we can test it in before we look to implement it in people.
And actually, sort of down that same line, some folks ask questions about really specific researchers' progress. So, we have questions about Denosumab, which you addressed, and bisphosphonates, and especially bisphosphonates and Denosumab together. Mm-hmm. Can you speak to that?
Yeah, no, that's also a really great question. So bisphosphonates act very similarly to Denosumab. They also are a medication that decreases the activity of osteoclasts or bone resorbing cells. What's been so disappointing about bisphosphonates is they just don't seem to work in FD the way Denosumab does. And the reason for that probably is, and this is speculation, is that, unlike Denosumab, which just targets RANKL, it's a small protein and all it does is it targets RANKL. Bisphosphonates are a lot more complicated. They have to get incorporated up into bone tissue to be able to work. And in FD, we don't have a lot of bone tissue. We mostly just have this fibrous tissue that doesn't have a lot of structure to it. So there is, I think, a real potential that if we give patients Denosumab and we build that bone within the FD lesions, you then might be able to follow it up with bisphosphonates and help maintain some of those benefits. We don't know if that's the case yet, though, and there are potential safety issues to using these medications together. They both have similar a similar side effect profile. They both increase risk of osteonecrosis of the jaw. They both could potentially increase your bone mass in non-FD bone. So we have to be really judicious. We don't want to over-treat patients with anti-resorptive medications. But I do think that going forward, this is something we're absolutely going to implement in our pediatric trial. And that's part of the reason we're using such small doses of Denosumab to make sure we don't overdo it with these osteoclast drugs.
And is that the bisphosphonate and Denosumab, is that something that's already happened in the mouse model studies? And that, or
not that I know of, but
okay.
I do think it's something people are working on. One tricky thing about the mouse model is I don't think anyone has yet shown whether or not they're a good model for this bone turnover rebound. They definitely seem to take up Denosumab and get similar treatment effects as people, but mouse bones at the end of the day really are quite different from human bones. So we're still trying to figure out if they're a good model for this big rebound of bone turnover. I suspect they probably will be, but I just can't say for sure yet.
Sure. Are there other drugs in development for treating FD?
I'm not aware of any other drugs that are at the stage where they're ready to be tested in a clinical trial. But one of the goals of having a mouse model is that you can do more detailed studies to look at what are some of the other signals that the FD cells are making. Is there something more convenient than rankle. The rankle pathway seems to be a great one to target. It's the very first time we've ever seen anything impact FD lesion activity, but it's really inconvenient. So I know people are looking at, is there a place earlier in the pathway or later in the pathway that we can target without running into some of these other problems we're seeing?
And then there was one question about regenerative research on the GNAS mutation. I don't know if that is just another way of saying gene therapy or regenerative medicine.
Yeah, I'm not quite sure what that would be referring
Okay.
to. Yeah.
It's possible that it's another way of addressing gene therapy, gene editing.
Yeah.
What about, so this is sort of more on the patient, the clinical side of things. What can you discuss new imaging that the NIH is using?
Yeah, I think the biggest advance over the past few years has been the sodium fluoride PET CT scans that I showed in the Denosumab part of the talk. So those are really wonderful imaging modality because they show us not only anatomically where the FD is, so you get a beautiful view of how big is the lesion, what bone is involved, what does it look like, but it pairs that with what we call functional imaging. So how active is the bone? How much is it turning over? What is its metabolism saying? So you really kill two birds with one stone using those scans. When we first designed this Denosumab trial, we had no idea if that was going to be a helpful type of imaging at all. But when we saw that the activity decreased so beautifully and that it decreased the same way the blood markers decreased, it showed us that this is a really excellent surrogate endpoint. So I'm expecting that this is going to be much more widely used in FD. A big downside of it is it does include radiation. So you want to be careful that you're not getting too many of these scans. One of the difficulty with imaging in FD is there's just no good way currently to see the bone without radiation. You can see other tissue without radiation, like brain tissue, soft tissue, heart, lungs. Those are all easy to see with things like MRI, but bone, you just have to include some radiation. So I am really hopeful that there may be more research into different imaging techniques that use less radiation. Also, faster imaging. One of our biggest challenges on our pediatric Denosumab study is that the kids have to lay still on the table for 30 minutes to get that bone scan. And some of them—
A long time for me.
Yes, some of them are quite wiggly. So we have them practice at home sometimes, laying still in the bed, getting ready for their scan. But I would love to make this easier for them.
All right. Actually, we have another question. We have lots of really great questions coming in about the Denosumab pediatric trial. How is that rebound effect thought to be less at small dose, and how is that being addressed in the trial?
Yeah, so the focus on maintaining safety through bone turnover rebound is a huge part of this trial. We have put a lot of thought and effort into making sure we are controlling the rebound as safely as possible. So I do think lowering the dose of Denosumab is very likely to decrease the size of the rebound. We will be using bisphosphonates to help blunt that rebound in the pediatric trial. So each kid will get at least 2 infusions. And then also what I think is a very important part of this trial is we have a lot of flexibility. We can give more bisphosphonates as needed. We can give more Denosumab as needed. We have the flexibility to do whatever we need to do clinically to make sure that rebound is controlled and made slower. One big lesson from our adult study is we found that sometimes you can help slow that rebound down if you actually, sort of counterintuitive, but if you give more Denosumab. So you let people rebound a bit, then give another dose of Denosumab, slow it down. So instead of having one big rebound that can be scary, you can have multiple small ones that are more controlled.
I can imagine as a parent, this being a thing of hope, but also really very terrifying. And I definitely appreciate you.
Yeah, I do think that's why I think it's so important we study this in a pediatric clinical trial, because there is no safer way to take a medication than on a clinical trial. We have so much regulatory oversight on this trial. We are being reviewed by multiple bodies. The safety is just really paramount. It's also a reason we're trying to target this trial to kids we think will benefit the most. Kids who already have a substantial amount of FD, who have already had an FD-related complication, because you want to make sure that the benefits of this drug, which I think could be huge, but also outweigh the risks, which are also substantial. At the end of the day, this is not going to be a drug for every kid with FD, but I think it could potentially really be super impactful in the right kids with FD. So that's really important to us.
And you know, just because we have so many questions about gene therapy and CRISPR, it sounds like that's sort of a similar balance right now. We're sort of watching the field, and when the risks and benefits hit that right balance, then we can get more excited. So it's not off the table. It's just farther down the line.
Yeah, there are so many genetic disorders that I think gene therapy could be a life-changing approach for. There are many disorders that are caused. So gene therapy works best if you have a single gene that causes the disease. And particularly a lot of these really devastating genetic diseases that can be fatal in infancy are often caused by single genes. So to me, that really seems to be the obvious place to start. You'd be willing to tolerate a lot more risk with a disorder like that than you would be in a disease like fibrous dysplasia, where we do have some ways of managing it. So that's what I'm planning to do, is really watch this field, see how it works out in some of these more life-threatening disorders. And then once it's worked out fully, if we have really reliable, consistent gene therapy, I do think FD could be a great disorder for it, because it is caused by a very consistent single gene. But if I had to speculate, I do think this is fairly far off, not outside my lifetime far off, but by a good 10 years off. But I would love to be proved wrong. Bring me back to prove me wrong one day, Tova.
I can't wait. I can't wait for that day. There are still so many good questions. I do want to make a note that we do get some questions about like specific cases and treatment plans. And this is not that webinar. There are great webinars and our treatment guidelines, which I'll be sure to put a link in the Facebook chat so folks have that. And you know, we want people to have their questions answered. So you can always reach out to us. Right now, though, we have a question about why did the NIH study fail to identify drug candidates? I think this is talking about those screenings.
Yeah, no, great question. We put so much work into that approach. It was disappointing. So we actually identified multiple candidates on our first run. So the way the screening approach works, it's very systematic. So you start with kind of and I'm just going to use metaphors here, but you do sort of a shallow screen on a huge number of compounds, and you see some possible hits. So what we used is we measured cyclic AMP response to these compounds, because that's kind of a downstream protein that gets made when G-alpha-S gets turned on. So we were looking for anything that can alter cyclic AMP. Maybe cyclic AMP went up, maybe it went down. We didn't care. Like, did it look like it was changing this at all? So we identified several, and I may be wrong on these numbers, but like several hundred compounds that seem to show potential. But then after that, you conduct a deeper screen. So maybe you do it in a kind of standard cell and see if it's impacting G-alpha-S there. Then you may move it up into a human cell that is much more, similar to the type of system we're trying to implement. And when we move to those further screens, the number of compounds that were actually able to alter G-alpha-S just got smaller and smaller, and eventually there were none that looked like they could feasibly be turned into a drug. But again, we were limited by our molecular library. We started with 300,000 compounds, and that seems like a lot, but with newer technology, you could potentially start with 1 million compounds. The library depends so much on what possibilities you could find. So I absolutely don't think that line of research is dead just because we didn't identify any with our screen. Every year we identify there are more potential molecular compounds to look at, and every year there are better screening techniques to look at them. Again, it takes from the time you find a possible compound till the time you have a drug you can put in a human's body does take a long time, probably at least 10 years. So I don't think this is anything that is going to be implemented in the very short term. But I would not be at all surprised if that becomes kind of the next hot area for FD treatment. I am hoping, as excited as I am about Denosumab, and as much as I think this could really transform lives for some severely affected kids, I do not think it is good enough. I would like a better, safer, more easier to use drug down the road.
We're behind you. I think I can speak for everybody on this call. Let's see. So there is a question. The clinical trial is ongoing right now. It sounds like Denosumab right now for children and burosumab for both kids and adults.
Yeah, so the burosumab trial is just for patients who have low blood phosphorus levels due to high FGF23. And then we do have our natural history study. All of our interventional studies are kind of enfolded in the natural history study. So patients participate in both. Okay.
And then the Denosumab trial in adults, has that been published yet? Is there hope of seeing that?
It is going to be available any day. It's already been accepted. So I check the internet every day for that trial. So very soon.
Great.
But, I do think we do have a version of it available online on what's called a preprint server. So those are articles that have not been fully reviewed by scientific peers and are not like official versions, but you can see all the results there.
And when that is out, I know we'll have a link to that on the FDMS Alliance website as well. We are coming up to the end of our time. Alison, I don't know what your time is like. Can you just speak generally, what's on the horizon, where can we, the community, be excited to look and where research focus is heading?
Yeah, so I really think there's a lot of reasons to be optimistic. I've been studying FD for a good 10 years now, and it has been so frustrating. I've spent many years watching kids have their lesions expand, watching people fracture and having nothing to offer. To me, the most immediate immediately exciting treatment is the potential for Denosumab and its ability to potentially change the quality of FD lesions. The most important short-term need in the community right now, I think, is figuring out how can we use Denosumab and who are the right patients. Again, it is not for everybody, but it is certainly for some. And the reason that's exciting is because it's here. It is approved. You can get it in your pharmacy. It has all the work to develop it, to test it, safety. All of that was done before in other disorders such as osteoporosis and bone tumors. So that's where I have the most short-term optimism. And long-term, I still have a fair bit of optimism about G-alpha-S receptor modulators. I think one thing that has really expanded our ability to look for those are these mouse models. Even 5 years ago, we did not have the disease models we needed in fibrous dysplasia, and now we have a good 4 mouse models. It's really expanded our ability to test things. So that's my medium-term excitement. And still say, although I think I did talk a lot of smack about gene therapy, that's my long-term excitement. I do think one day that's where we're headed.
Awesome. Alison, are you a hard stop in the next few minutes?
Yeah,
pretty soon.
I can do maybe like 2 more minutes.
Okay, sure. I again want to make it an opportunity for anybody who's here, joined us on Zoom. We do have some other questions, some really great questions. I do hope you will email us here. I'm going to put up this has been really a wonderful, wonderful program. Thank you so much, Dr. Boyce, for your insight and your expertise. Thanks also to our supporters, including Ionis and Ultragenics, and especially our Charlie's Angels monthly giving circle. We have that post survey that we really would love your feedback on. So I will put that in the chat and also on Facebook. And then finally, I do see, Alison, we've got another question that came in the chat here.
I could do one more. Let's see.
You can do one. Okay, let's see. We've got, what will rankle inhibition not fix?
Good question. So rankle inhibition, I think at the end of the day, seems to be very effective in making these FD cells less active. However, I do not think they're going to fix an FD lesion that's already formed. A lot of the complications with FD, although I talked a ton about cells, are structural. So once you have a bone that has formed in a way where it's incorrectly, so it's either too expanded or it's bowed or it's too large, there's no way to put that back in the box. We can make the lesion less active. We can make it stronger, so it's less likely to fracture. We can stop it from expanding anymore, but we can't turn it back into a normal bone. So I think that is unfortunately a big disadvantage, and is one reason why we're really focused on, children are not going to be the only patients who will benefit from denosumab, but I do think that is where we'll probably get the biggest bang for our buck, is stopping those FD lesions from getting worse once they've shown up.
Great question. Again, if your question didn't get answered, please do reach out to us through the survey, through our contact us page. We're really excited about our upcoming events. In February, we have fdmas Global Awareness Week with our first ever art contest. We're really excited about that the Patient Advisory Council has put together, as well as a community social. And then later this year, we have our in-person fdmas community conference. We're so excited to be back in person in the Silver Spring, Maryland area, September 8th through 10th in 2023. We'd love for you to join us. We'd love for you to be involved. So definitely, we hope you connect. And thank you so much, Alison. And thank you, everyone.
Great. Thanks all. Take care.
Well.